Good afternoon, everyone, and welcome to Myositis Support and Understanding. We're also known as MSU. We're happy to have you join us on Rare Disease Day today. My name is Jerry Williams, and I am the founder and president of Myositis Support and Understanding. And I've been living with what my doctors now think is dermatomyositis rather than polymyositis with skin lupus since 2003. MSU is an all-volunteer nonprofit organization working to improve the day-to-day lives of myositis patients and caregivers. And one of the ways that we do this is through education about clinical trials and research to help us all better understand and get the information we need to participate in clinical trials and other types of research. So join me in welcoming Dr. Mary Katherine Farmer, a rheumatologist and senior medical director at Kezar Life Sciences. Today, she's going to introduce Kezar, KZR-616, and the PRESIDIO clinical study. Having worked with many of the major pharmaceutical companies, including work on several medications that have been approved for different diseases, Dr. Farmer has many years of experience in clinical trial development. And in mid-2019, she took a once-in-a-lifetime opportunity to join Kezar Life Sciences to run the Myositis Development Program. Good afternoon and welcome, Dr. Farmer.
Hi, Jerry. Hi, everyone. Thank you so much for the invitation to talk to everyone today. I'm speaking from my home office, so I apologize if there's any noise from my painters or construction crew that are here. But again, thank you so much. I'm going to put myself off video and then so you can see my screen. And then I appreciate your attention. I'm going to try to explain everything so that you can understand it. And at the end, we'll have plenty of time for questions. So again, thank you so much, Jerry, on behalf of MSU for inviting me and Kezar to talk about our program. Today, I'm gonna introduce you to Kezar. I'll talk a little bit about inflammatory myopathies. I'll give you the science behind KZR-616 in words that you can understand. And then also tell you why we think that KZR-616 has the potential to treat poly and dermatomyositis. I'll give you a little hint about the clinical data so far from humans in the clinical trials to date, and then talk about the PRESIDIO trial on PM and DM. But before I do that, I'd like to tell you a little bit about my story first. So you're probably wondering what my connection to myositis is. A lot of us that work in myositis have a connection. If you read my bio, you'll see that I've had dermatomyositis for about 17 years. When I was training as a resident in internal medicine, which is a very, very stressful time. You have a lot of responsibility, you don't get any sleep, you have a lot of things to do for a lot of people and making sure everybody's okay. But I started to feel bad, a lot of fatigue, and I developed this kind of strange rash on my arm. Went to a few doctors, said, you have fungus on your arm, you need to use this, you need to use that. But I convinced a rheumatologist who was visiting from another institution to do some basic blood work. So when my liver enzymes, or AST and ALT, came back in the thousands range, which is really, really high, I was questioned ad nauseum about my alcohol intake. And I didn't really drink much then or now. And so long story short, I ended up just begging for a CPK to be drawn. And that was 50,000, which led down the road to my diagnosis. Underwent an EMG, which was completely normal, had a muscle biopsy, which was classic for dermatomyositis. I've been lucky. I was put on high-dose steroids and tapered off, take, immunosuppressants to kind of keep everything at bay. I lead a pretty full life. I have a lot of us deal with new normals, and, I have new normals every now and then, and I'm okay with that because I really, love what I do for a living, and I work with a great team of people, and I feel fortunate that we have, medicines that work somewhat well for some people, but we need new treatments, as I think we can all agree. So again, thank you so much for joining us today. So who is Kezar? This is Kezar. We are a fun, loving group of hardworking, really bright people. We love Halloween, and this is a picture from Halloween. And this is Kezar. You can see everyone is smiling. These are true, genuine smiles. Again, we love what we do. And we really have one focus, and that's to help bring new medicines to diseases that need better treatments in autoimmunity and cancer. So you're familiar with DM and PM, but as you know, they're chronic autoimmune diseases characterized by inflammation of the muscles and associated tissues. Back when, this slide is data from years ago when we really knew about dermato and polymyositis. So these are also called the idiopathic inflammatory myopathies, meaning that idiopathic means we don't know where they come from. Inflammatory means that inflammation causes the problems. And myopathy, something's wrong with the muscle. So we don't really know what causes them, although we know it's probably a combination of something in the environment and your genes. Muscle weakness and fatigue are common. People with dermato get a skin rash and other skin manifestations. And as you know, other diseases can be affected too. Some people get interstitial lung disease. Some people, especially children, get horrible GI symptoms like horrible reflux and problems with their intestines. And then in the heart, you can get inflammation, rhythm problems, etc. The Bohan and Peter criteria from 1962 are listed here, which I won't talk about, but just to show you that they're there. So the idiopathic inflammatory myopathies are comprised of 6 different diseases. The ones starred in red are the ones that we're allowing into the PRESIDIO study. So dermatomyositis, amyopathic dermatomyositis, which is basically the skin findings of dermatomyositis without the weakness. A lot of these patients go on to develop the weakness, but not everyone, especially if they're treated. Polymyositis, sporadic inclusion body myositis, and then the new kid on the block, immune-mediated necrotizing myopathy. And this is called a myopathy, not a myositis, sometimes, although you'll see both terms used. This is more of a muscle dysfunction. There are varying degrees of inflammation on when you look at it under the microscope on a biopsy and MRI. But that's not a compelling, it's not really a prominent feature. These diseases can occur in childhood, and they're called juvenile dermato and juvenile polymyositis. And then there are some diseases that include myositis as a feature. One is antisynthetase syndrome, and this is a cluster of symptoms that include interstitial lung disease, which causes inflammation and scarring in the lungs. Having poly or dermatomyositis is a requirement. A lot of folks have polyarthritis, which is arthritis or inflammation of multiple joints. Raynaud's is a common feature, causing color changes in the fingers, mostly when exposed to cold or shopping in the frozen food section of the grocery. And mechanic's hands, which causes some fibrosis, and which is scar tissue and trouble with the hands. And then there's also mixed connective tissue disease, which has myositis or can have myositis as a prominent feature. So these diseases are associated with certain antibodies in a lot of cases. An antibody is a protein that your body makes. And sometimes these antibodies can be what we call pathogenic or cause problems in the area where they deposit. So here, over the past few years, we've made a lot of progress as in medicine in finding new antibodies. So in the middle, you'll see DM. DM, we have several antibodies that are associated with DM. I'm an anti-MI2, I'm certain. I've never had it done, but I certainly act like it. I get the Shaw sign around my neck. I get the arthritis. I respond well to steroids. And my skin rash is pretty prominent when it is. And then there's NAM, which is necrotizing autoimmune myositis or immune-mediated necrotizing myopathy. And with those, we see a lot of anti-HMG-CR antibodies. And then the overlap antibodies, over on the left-hand side, we see anti-Jo1 with antisynthetase syndrome, and then some others in overlap that are seen. For instance, anti-DNA antibodies are seen mostly in lupus, but can be seen as a myositis-associated antibody. So the MAA is myositis-associated antibody. So those are associated with disease, but the myositis-specific antibodies are specific for disease. So if you have an MSA or a myositis-specific antibody, you likely have myositis or myopathy. So then how do we treat these diseases? As you probably know, steroids are usually or often the first-line treatment for a lot of autoimmune diseases, but including idiopathic inflammatory myopathies. So steroids reduce the inflammation that causes the muscle weakness, and it also suppresses the overactive immune system. We love steroids because they work quickly and they make us feel good, but they're not without risk. You are probably familiar with some of the long-term problems. They can cause cataracts, they can cause heart disease, they can cause brittle bones, they can make you more susceptible to developing diabetes. So the goal is to get the steroid dose down as quickly as possible. And sometimes we have to do this with introducing another medicine called an immunosuppressant. So these are what we call steroid sparing, meaning that we can get the steroid dose down when we use these oftentimes. So some of these medicines you've probably heard of: azathioprine, methotrexate, tacrolimus, mycophenolate mofetil, leflunomide. And then both steroids and immunosuppressants, or the two together, work for some people or a lot of people, but they also have side effects that can be serious. So these tend to be immunosuppressing, meaning that it calms the immune system down. By doing that, it can make you more likely to develop severe infections. So I think we're all in agreement that we need better treatments. And it's really exciting to be working in myositis right now because there are a number of companies who also have this as a focus. And as a myositis community, we're really excited about this. So now I apologize, and I'm gonna take you back to high school biology for just a minute because I want you to leave my talk today understanding how KZR-616 works. So the body is comprised of different organs. Each has a different function. So this is a section through the chest and abdominal cavity. And they're different organs: the lungs, the heart, the liver, the stomach, the intestines, etc. So just for example, the liver is an organ that is responsible for detoxifying things that you take in or that you're exposed to. So it takes things that are bad for the body or harmful, and it changes them through a series of chemical reactions, and it makes them so that you can excrete them out either through sweat, urine, or through bowel movements to get rid of those toxins. And then each organ is composed of millions of cells, which are sort of the building blocks of an organ. And each cell is composed of different functional units called organelles. Each organelle has a specific function. So you can see here sort of at the top right, the mitochondrion is what we call the powerhouse of the cell. So this part of the cell helps your body make energy from different things lying around by putting them together, taking them apart, and changing them from one form to another. So organs in your body are composed of cells, and then cells are composed of organelles, each of which has a different function. I'm going to talk about proteasomes now. And these are directly involved with the way KZR-616 works. So a proteasome is an organelle, and it's a protein found in cells in the body. Every cell in the body has one type of proteasome or more. So the proteasomes break down damaged and unneeded proteins. It's sort of like the garbage disposal of the cell. So they're part of the major mechanism by which cells regulate proteins and destroy damaged proteins. So it's like they tag these abnormal proteins for removal by sticking a protein called ubiquitin onto them. And then they're broken down into smaller pieces called peptides. And then the peptides are their small protein fragments. Then those are broken down into amino acids, which are the building blocks of protein. So now the building blocks of protein, or the amino acids, are able to be synthesized or made into bigger units and sort of recycled, if you will, to make new proteins that the body needs. So the proteasome is an organelle found within all cells. So the slide I showed here, it didn't show the proteasome, and part of that's because proteasomes are relatively new to, were discovered not until the 1970s, and so they've just become, people are starting to recognize them more. And here you see the picture of the proteasome. So that's the general proteasome, but then there's something called the immunoproteasome. So this is a type of proteasome that is expressed in the immune system cells and also where inflammation occurs. So proteasomes are everywhere, and then immunoproteasomes are in the immune system, and then in places where inflammation occurs. And inflammation in the body can occur when there's something wrong. We talked about antibodies earlier associated with myositis. antibodies can go to normal tissue, normal cells, normal organs, and they can cause a set of reactions in the body that causes inflammation. So if we can cut down on the antibodies or reduce the antibodies, we can theoretically help heal the problem or heal the disease, heal the inflammation, get rid of the inflammation, etc. So here you see on the left, the constitutive proteasome is in the heart, lungs, etc., whereas the immunoproteasome is at sites of inflammation and in the immune system. And there's several types of cells listed there. The B cells are the ones that secrete antibodies. So it's all starting to, you see where I'm going with this, is that we have different antibodies associated with different myopathies or myositis conditions. The antibodies are secreted by B cells, and the immunoproteasome is within these B cells and other immune system cells, so that if we could find a way to block the immunoproteasome or modify it, we could potentially treat diseases caused or seen with inflammation. So by targeting the immunoproteasome, we represent a unique approach to treat inflammatory disorders. So proteasome inhibitors such as bortezomib, which is approved for treatment of multiple myeloma, which is a bone cancer, we use animal models, which are sort of the way we look at disease before we treat patients with a drug. We often use an animal model to show whether or not we think it's going to work in humans. So we have evidence that bortezomib it works for animal models of lupus and lupus nephritis. Bortezomib, which is a dual proteasome inhibitor, dual meaning it hits uses the constitutive proteasome and the immunoproteasome, dual meaning two. This has been successfully used in the treatment of hard to treat autoimmune disorders. The problem with this is while it works to treat the disease, there's problems with side effects, including prolonged fatigue, peripheral neuropathy, which is numbness and tingling, in your hands or your feet, and also problems with different types of blood cells, which can be really serious. So by selectively inhibiting the immunoproteasome with KZR-616, for example, we can replicate the anti-inflammatory activity of bortezomib without seeing the similar side effects in animals. So there are a number, 20 plus peer-reviewed scientific articles, which support using the immunoproteasome inhibitors for the treatment of autoimmune disease. A peer-reviewed scientific article is the most rigorous kind of article. It means that people who are experts in the field say that the paper has merit or should be published. So this is really exciting because this is a really good foundation for KZR-616. So KZR-616 is the product of long-term research and development efforts. So I mentioned that the proteasomes were discovered in the 1970s. In 2004, a Nobel Prize was awarded for proteasome discovery, some almost 35 years later. Proteasomes entered clinical trials about 1999, and 4 years later, Velcade or bortezomib was approved, the first proteasome inhibitor approved, and that was for multiple myeloma or the bone cancer. The first selective immunoproteasome inhibitor was discovered in 2009, and then Kezar was formed. KZR-616 was nominated for clinical studies, and then the next year, the first clinical trial with KZR-616 was initiated, and then PRESIDIO study was launched last year. The PRESIDIO study is the study in myositis. So we believe that KZR-616 has the potential to treat severe autoimmune disease. So our development program right now is looking at 5 diseases. They're all named for San Francisco neighborhoods, which we're very proud of. The one for blood disorders is called Marina. Lupus nephritis and lupus is called Mission. And for dermatomyositis, including necrotizing myopathy or immune-mediated necrotizing myopathy, is called PRESIDIO. These are all phase 2 trials, which means they're fairly early in development. And they're smaller than if we were using them to try to get drug approval. So the really exciting, what gets me excited when I wake up every day and excited about working on this program is that earlier when I said that we use animal models to get an idea of if something will work or not for humans, we have fairly new data showing that, that inhibiting immunoproteasomes can improve muscle function in a mouse model of polymyositis. So I'm just going to tell you a little bit about this. I won't talk much about the graphs at the bottom, but just to say that this is a CIM model, which is a C protein-induced myositis. So how this works is that you give mice a shot of a muscle C protein. This causes a myositis in about 13 days in the animals. You measure their grip strength before they're treated, and you do this with a specialized contraption that's really cool. And then you check them afterwards, and then they develop weakness. And then evidence when you look under the microscope at their muscle of inflammation and necrosis or muscle cell dying that we see with polymyositis. So then if you treat these animals with ONX-914, and let me tell you about ONX-914 real quick. So ONX-914 is what's called an analog of KZR-616. It means it's just an earlier form. It's very similar to and acts the same way that KZR-616 does. So when you start treatment with ONX-914, it improves muscle function and then reduce muscle damage when you compare a CIM mouse with given the muscle C protein with one that you also give ONX-914 to. You see markedly less inflammation is shown at the bottom of day 28, and then enzyme levels with creatine phosphokinase as well. The levels are reduced. So that's really, really exciting. So what about the human data? So KZR-616 has been studied in about 100 healthy volunteers and 33 lupus patients. In healthy people, there were several doses that were well tolerated, and doses that are higher than the ones we're using in the PRESIDIO study for myositis. And we haven't seen the side effects that are normally seen with the dual proteasome inhibition, such as bortezomib. So we haven't seen these blood cell problems, or the peripheral neuropathy, or the severe fatigue. And then in patients with lupus and lupus nephritis, which is kidney disease related to lupus, so far there have been 33 patients treated. And in terms of looking at how well this has worked against lupus, we looked at seven different ways to measure disease activity and how patients do, including a composite of a lot of different functions, and then something called CLASI, which is the Cutaneous Lupus Area and Severity Index, which is basically a way to look at lupus skin disease by how red it is and how much of your body it covers in those patients who have skin manifestations of lupus. Not everybody with lupus gets skin disease, but in those who do, we found that after 13 weeks of treatment, that the skin improved significantly in those patients who had that. Also, tender and swollen joint counts got better, and patients and physicians reported that they felt better or that their patients felt better. So interestingly and excitingly, the majority of patients that took KZR-616 for at least 1 month, which was 4 doses, had some sort of improvement in their disease activity. So that gets us really excited. And then we're also excited about KZR-616 for other autoimmune diseases. So selective inhibition of the immunoproteasome occurs without the side effects seen with dual proteasome inhibition, like with bortezomib. We have encouraging data from laboratory research in animal models of autoimmunity, including polymyositis with this mouse CIM model. Improvement in the CLASI score, skin scores in lupus patients in what's called our mission study, lupus nephritis. This suggests that the skin findings in DM might improve with KZR-616, because when you look under the microscope, patients with DM have very similar appearances to patients with skin lupus. And some of you probably were misdiagnosed with lupus based on a skin biopsy. And then after time went on and you developed muscle disease, that diagnosis was changed. So our long-term goal is to establish KZR-616 as a standard of care for the treatment of severe autoimmune disorders, including DM and PM. And right now, KZR-616 is the only immunoproteasome inhibitor in clinical trials right now for PM and DM. So this is our website for the PRESIDIO study. The web address is there. Please check it out when you have a minute. It's pretty exciting. We have some good information about poly and dermatomyositis, as well as about KZR-616. A few hints about who might be eligible, as well as the study locations and a way to contact us. So this, our phase 2 study, PRESIDIO, is actively enrolling poly and dermatomyositis patients. So over on the left-hand side, it's either a purple or a gray box. Adult patients with DM or PM, N equals 24. That means that we're looking for 24 patients total. We hope to have about half with dermatomyositis and about half with PM or necrotizing myopathy. So what happens is that people are either randomized, which means assigned without knowing, to start treatment with KZR-616 and then go to placebo, which means that there's no active KZR-616. Or people start with placebo and then go to KZR-616. It's a weekly injection, so it's a weekly injection for 13 weeks, followed by the placebo period or the treatment period, depending on what you start out with or what your initial treatment assignment is. And then there's 8 weeks of follow-up. So during this time, at study visits, what happens is that you have a number of what's called patient-reported outcomes that you do on a tablet or on your phone. You go into the study center. There are some assessments done by someone at the study site to look at your strength and your function and how well you do things. And then there is some blood work drawn to look at your blood levels of different things. Then you receive the injection, and then you come back the next week for another injection, or you have a home health service do that, and we'll talk about that in a minute. So what's unique about PRESIDIO? So everyone gets KZR-616. So most studies have some or most of the study participants receive the study drug, but for PRESIDIO, everyone gets the chance to receive it. That's really exciting. It's called a crossover study. So study sites can utilize home health services to administer the weekly injections for most of the study visits. Now, some study visits have to be done on site because they involve checking your function and your muscle strength, et cetera, and drawing blood. So those are done at the study site. But for all the other ones where there's just an injection and a few questions, you can, if your site decides to utilize this, this can be done. So it's really helpful for those who work or who are physically limited. I don't know about you, but me, I have some physical limitations. So it's harder for me to get somewhere like that than it is for somebody without myositis. So the nurse comes to you, and then we're in close contact with them, and they pass on any information we need to know about. And then doctor visits, assessments, any testing, and KZR-616 are free to the patient. And then you as the patient have frequent contact with your study site. So then what everybody wants to know, like who might qualify for this study? So if you have DM or PM, which includes immune necrotizing myopathy, we accept antisynthetase syndrome as well as secondary Sjogren's. Sjogren's is a constellation of symptoms associated with a lot of autoimmune diseases that causes dry mouth and dry eyes. People with stable interstitial lung disease are also possibly eligible. And if you use an assistive device like a walker or a rolling walker or a cane, that doesn't mean you can't participate, because we know that these are, a lot of people with myositis need these. So the type of patient we're looking for has active disease and some muscle weakness. Both are required. So active disease is shown by an active dermatomyositis rash in patients with DM. Active disease in the muscles shown by a recent MRI, an EMG, or a muscle biopsy, and recent was within 6 months. Elevated CPK we often see in patients with immune-mediated necrotizing myopathy, and for those patients that's acceptable to show active disease. And then, so that's the active disease part. And then the muscle weakness part is determined by standardized muscle testing that somebody at the site will do. And there's no absolute requirement to be taking medications for myositis, but the following are permitted during the study. So up to 20 milligrams of prednisone per day, and then 1 steroid, which is prednisone or another steroid, 1 immunosuppressant, which can be azathioprine, methotrexate, mycophenolate mofetil, tacrolimus, and 1 anti-malarial for DM. And that includes something like hydroxychloroquine or Plaquenil. So up to 20 of prednisone, 1 immunosuppressant, and 1 anti-malarial. And this is a screenshot from the PRESIDIO website. So the other thing I'd like to mention is that when we go to seek a drug approval with the FDA and other regulatory authorities, after the phase 3 trials, et cetera, we seek a certain designation, if you will. So what we're likely to seek at this point is people with active disease who have been either treatment resistant to standard of care therapy, which would be steroids and or immunosuppressants, or they can't take them because they've had side effects. So that's also factored into who qualifies for coming into the study. And then what types of patients would not qualify for PRESIDIO? So sporadic inclusion body myositis and juvenile onset myositis, cancer-associated myositis, myositis in overlap with another connective tissue disease. But I'm gonna caveat that because there are a lot of people walking around with a diagnosis of myositis and rheumatoid arthritis. Now we know that myositis is frequently associated with polyarthritis, which is inflammation of multiple joints. So sometimes someone will assign a diagnosis of rheumatoid arthritis in your chart, either to get a medication paid for by insurance or just to make it easier to get certain things done. But if you don't truly have rheumatoid arthritis and myositis, then you might be eligible. People with normal muscle strength are not eligible. And those with moderate to severe muscle or lung damage are not eligible. But I feel that our inclusion and exclusion criteria, which is the way we decide on who can come into the study, I feel like they're very generous and allow for a lot of potential different types of people to come in. So then medications that are not permitted include prednisone more than 20 milligrams, more than one immunosuppressant. So a lot of people take azathioprine together with methotrexate. We don't allow the combination to come into the study. If you take Acthar gel, you can't take it during the study, but if you've been exposed prior, you can. And then a lot of people take immunoglobulins, IVIG or subcutaneous IG. These are not allowed during the study, but they're allowed before the study starts. So if you've been exposed to those before, that doesn't mean you can't come into the study. So we currently have 8 sites up and running in the US that can enroll patients, and we have more coming. We've shown, this is from our website, these are the sites, and we currently have 2 patients enrolled and several more who are in screening. So it's really exciting. There's been a lot of excitement around this study, and we're really excited too. And I'm sure that comes through while I'm talking. So please check back frequently for updates on sites, etc. So I'm going to stop there, and I'm going to open it up for questions. And I think that Jerry and his team have been getting the questions ready for us.
Dr. Farmer, thank you so much. That was a wonderful presentation, very informative. So we thank you for taking the time to kind of walk through all of that science and kind of bring it all together to give us a better idea. I know one question that jumped out immediately was Rituxan. Is Rituxan something a patient can use?
You can have rituximab prior to coming into the study. It must be 6 months or more. But yes, it's allowed. We know a lot of patients take it too. So for patients who have had it within a certain time period, we check a certain B cell, we check a certain count in your blood. And then based on that number, you're likely to be admitted to the study if everything else is okay.
I really, really appreciate the excitement, hearing it in your voice, and then, you know, knowing that you have DM and are doing this. It's incredible, and hearing the hope in what it sounds like it has the potential to do is really exciting. So I just appreciate you giving us this presentation.
Oh, thank you so much. That means a lot. I left a very stable job at a very large company to come run the program. The science, I met Chris Kirk, who's our Chief Scientific Officer. I met him in a meeting a year and a half before I came to Kezar, and I was blown away by the science. It made so much sense, and I couldn't. There's no way I was going to turn this down. So we're very hopeful too. And thank you so much for your comments.
It almost seems like almost like a more targeted, focused way of treating as opposed to Rituxan, which maybe is a little more broad, where it.
Your analogy is a very good one. It's similar in that you're right, it's targeted. So we know that the drugs work that aren't targeted, but when we target, we expect and hope that they will work better. And that's why, that's why we're doing the clinical trial, because we'll be able to get data on real patients. And, you know, we're looking for several different types of patients, essentially DM, PM, and immune-mediated necrotizing myopathy. So we may get data in 3 different types of myositis, which would be really exciting.
Just a quick question for myself. For those that do have immune-mediated necrotizing myopathy, you don't require one of the antibodies, the autoantibodies, SRP or HMGCR, to confirm that, just the CK level?
Well, we don't require it, but what we found so far is we've had two patients that have screened or are in screening right now, and they've both had positive antibodies. But what we do with this study is a little different than other studies. We have what's called an adjudication committee, an adjudication process. So every patient who is screened for the study, you know, has all the blood work, all the testing, et cetera, I review that. And then based on that, it's either obvious that you completely are eligible, or it's obvious that you're not eligible, or there's a middle ground where you may or may not be eligible. So in that case, I sometimes I'll ask for more information from the site, like, you know, additional EMGs, et cetera. Sometimes we may ask for additional workup, like for somebody with polymyositis that's not immune mediated, that doesn't have an antibody, we may ask for like that an MRI be done or an EMG, et cetera, or biopsy be done, so we can see what that looks like. But then I pass that information on to several experts in myositis, and then they review it, and we come up with a consensus about whether or not you're eligible or not. So it's a very data-driven process, and I think we have it down to we're getting it down to a really nice way of kind of looking at everyone the same way and making sure we get the right patients into the study.
I've got necrotizing, and I can tell you right now, I won't qualify for the study because of how often I do get Rituxan, but I do know some people around my area. The question is, are you guys going to have any study places in the Northwest? Because the closest one to me is California. That's one heck of a trek.
We started with targeting myositis centers. Then we added a number of other sites, a lot of neurology sites that do clinical trials that had experience.
Hi, Jason. This is Kirthi. I'm the clinical trial liaison with Kezar. And I think your point is well taken, and it's something that we're aware of in terms of where our sites are and where we would like to see more additional sites open. And we are certainly still exploring those options right now. And I will get back to you guys as we open more sites, and particularly as we open more sites in the Northwest. I'll make sure that's communicated to you.
Yeah, that's great, because I'm noticing that there really is not a lot around here. You either live on it's either the East Coast, the Midwest, or California.
No, and that's I mean,
tucked up here is we're kind of fishing here for something.
No, and I think you're right. Unfortunately, the most well-known myositis center in that particular region is really in California. But we're working to identify trial sites.
We do have a question from Mary in the chat section. Overlap patients are excluded from this study, but might they still benefit from the new medication if approved in the future? And are they excluded because of confounding?
That's a great question, and I can tell you the answer. The answer is that we talked about that a number of times, and what we decided was that with only 24 patients, we really needed a more homogeneous or more pure population. And since we're allowing three different diagnoses into the study, we figured that we would not allow that now. But for phase 3, we're going to readdress that, because we do feel that patients with overlap syndrome could benefit. It's just that when somebody has, say they have lupus with myositis overlap, sometimes the lupus overshadows the myositis. So that would be a tough one to convince me that we should do that, but it's not unheard of. So we will have, I'm sure, multiple discussions about that as we get ready for phase 3. But thank you for your question. Thank you so much for your attention and joining us today. Again, it's been a true honor speaking on behalf of Kezar and KZR-616 on this very important day.
Yes, and we appreciate your time joining us not only on a Saturday, but on Rare Disease Day itself. As a physician. Right.
Yeah.
We appreciate the time that you took, and we thank the entire team at Kezar. You guys are wonderful to work with, and we thank you for that, and the educational opportunities for patients at the same time. So thank you again, Dr. Farmer. Thank you, Katherine. I appreciate you joining us today, and we hope everybody enjoys the rest of your Rare Disease Day.
Definitely.
Yes.
Thank you.
All right. Thank you so much. Bye-bye, everybody.
For more information about Kezar Life Sciences and the PRESIDIO Phase 2 study, visit kezarlifesciences.com/PRESIDIO study and via email at clinicaltrials@kezarbio.com. Thank you again for joining Myositis Support and Understanding with Kezar Life Sciences. At MSU, we're working to improve the day-to-day lives of myositis patients and care partners. Learn more about the work we do, including information about clinical trials, by visiting our comprehensive myositis website, understandingmyositis.org.